Ngalakin Khadijan Nyunga Boja.
Hello everyone. Today's lecture is going to look at chloroplast structure, electron transport, photophosphorylation, and ATP synthesis. Now, before we get started on today's lecture, I'm just going to tell you that we're moving through some of this information quite quickly because I have compared my slides with what you saw last year in BIOC3005, and some of the information is essentially the same. So I'm going to assume that that is knowledge that you already know or that you can build on and that you'll remember as we talk through the slides. And I'll point out some differences, and then of course we're going to build on the information that you learned in BIOC3002 last year in this particular lecture. So you'll see the parts that are new information. So as usual, there's some reading from the textbook, again, background information, almost assumed knowledge for some parts of the lectures, but it should help you out. And then also learning outcomes, where you can take those learning outcomes and shift them into questions and quiz yourself on the information. So when we think about photosynthesis, we actually have to think about two sets of reactions, and those are the light reactions and the carbon assimilation reactions. They're illustrated in the cartoon on the right-hand side of the slide. Today's lecture is focusing on the light reactions of photosynthesis. All right, that's where we see ATP being made, reducing power in the form of NADPH being made, and also oxygen being generated. And that, again, that's illustrated in the cartoon, where we have sunlight hitting the light reaction mechanisms, the machinery in the chloroplasts, and then oxygen being released, NADPH being produced, as well as ATP. And those products of the light reactions are then used in the carbon assimilation reactions that we'll talk about in the next lecture. So the essence of the light reactions and the carbon assimilation reactions is to convert light energy into chemical energy, and that chemical energy is in the form of reduced organic compounds. And essentially all life on the planet is dependent on this process. I mean, we're certainly dependent on plants because we eat them. If we eat meat, then the animals that we eat plants. But we're dependent on plants in other ways for fiber, for example, cotton, in many of our clothes. Also even wool, right, in our jumpers, because the sheep eat the plants. And in addition to that, wood, building materials. So no matter what part of our lives we look at, plants are incredibly important. And that's not to even go into how many people take pleasure in having plants around them and enjoy gardening and all the rest of it. So it's a very simple reaction that plants do on paper. Carbon dioxide and water, okay, with some light, leads to the release of oxygen and the production of carbohydrates. Okay, looks simple. Actually, it's quite difficult to do in the lab, but plants do it every day, all day, essentially, except when it's dark, and it's easy for them to do. It's also worth noting that oxygen is a byproduct of photosynthesis, and yet we're dependent on oxygen as are other organisms. So where do the light reactions take place? Again, this is a little bit of a review, but if we focus on the lower right-hand side of the diagram, then we're looking at the chloroplasts, where the light reactions happen. If we're talking about the prokaryotes, the cyanobacteria that do photosynthesis, then the reactions take place in the photosynthetic membranes in those unicellular prokaryotes. But in algae and in plants, whether we're talking about seagrasses or we're talking about land plants, the reactions take place in the chloroplasts. And the chloroplasts are surrounded by two membranes. So this should sound familiar because it's very much like what happens in mitochondria. The outer membrane is permeable to ions and small molecules, whereas the inner membrane is impermeable to these molecules. and instead has lots of transporters, so that anything from the cytosol or cytoplasm that has to get to the inside of the chloroplast has to go through a transporter on that inner chloroplast membrane. And then once inside the chloroplast, we see a third membrane system, and that's known as the thylakoids. And the thylakoids can be stacked, as shown in panel D of the cartoon, and the stacking leads to the membranes folding on itself, and where the membranes have folded, there's a space which is known as the lumen. So the stacks of thylakoids, the grana, but there's also these single thylakoids that are known as stromal thylakoids that connect one granum to the next granum. Okay? In the thylakoid membranes, that's where we see the photosynthetic pigments. We see all the light harvesting machinery there, as well as the ATP synthase. And something to put in the back of your mind for a minute, because we'll get back to this later in the lecture, is that there are lots of unsaturated fatty acids in the thylakoid membranes, which makes these membranes very fluid, allowing the complexes sitting in the membrane to move laterally within the plane of the membrane. And then inside the chloroplast, the part of the chloroplast that's not taken up by the thylakoid membranes, that's known as the stroma. That's where we see the enzymes for carbohydrate production that we'll talk about tomorrow. But it's also where chloroplast DNA is found, because remember, chloroplasts still have some DNA. Much of it's been transferred to the host cell nucleus during the endosymbiotic process, but some chloroplast DNA still remains in the organelle, and all the genetic machinery that's there for that chloroplast DNA to work or to do its job is there. So for transcription, for translation of the RNA that results for the replication of that chloroplast DNA, all of that machinery is also in the stroma of the thylakoids. Sorry, the stroma of the chloroplasts. So with respect to photosynthetic pigments, a word that you might not have heard or used too often is chromophores. So either word works, photosynthetic pigment as a phrase or chromophores works. And remember, these are molecules that have alternating single and double bonds or single electrons. And they strongly absorb visible light, which is of course what plants are being bombarded with. So something to remember about the chromophores is that they're always associated with proteins. They're not just there in a fluid, environment in the chloroplast, but instead they're associated with particular proteins, and they're organized in particular ways in the thylakoid membrane to optimize light harvesting. The chlorophylls are very noticeable, very in their structure, in the sense that they have a porphyrin ring and long phytol tails. So both chlorophyll A and chlorophyll B are shown in the diagram. And hopefully the ring structure looks a little bit familiar. It kind of looks like a heme group that we saw with the cytochromes. And instead of iron in the middle, it's magnesium atom in the middle of the porphyrin rings of chlorophylls. The two chlorophylls differ by a single place, and that's at the ring of 2 of the structures. And so in chlorophyll A, it's a methyl group, and chlorophyll B, it's a formal group that's bound to ring 2. And then on the lower right-hand side is a structure of beta-carotene. Again, those alternating single and double bonds, allowing electrons to be moved through those structures, or sorry, allowing, better to say energy at this point, better to say energy being moved through those the structures. And beta-carotene is an accessory pigment found in the leaves of land plants. And it's also, of course, the compound that gives carrots their orange color, but it does work in photosynthesis as well. So when we look at what happens when these chromophores absorb light energy, then we see that 4 different fates can be described for that energy. A photon, okay, so a packet of light energy, is absorbed by a chromophore. And when that happens, that chromophore, that pigment protein, has in it electrons that are lifted to a higher energy orbital, and the pigment molecule is said to be in an excited state. Okay, so the electron goes to a higher energy orbital, the chromophore is excited, and that's what's shown in the top part of the diagram on the right-hand side of the slide. When that electron has moved to a higher energy orbital, the pigment molecule, okay, is known or is said to be unstable. The electron will return to its lower energy orbital, meaning that the pigment molecule will return to its stable ground state. Now, there are 4 different ways that the pigment molecule can return to the ground state. In one way, okay, one of the scenarios, the far left-hand side, is through the dissipation of heat. Okay, so through the electron moving from the higher orbital to a lower orbital, the motion through the movement through the atoms in the pigment molecule give off the energy as heat. The energy may also be given off as fluorescence. So when a pigment molecule gives off light energy at a lower wavelength than what it absorbed, that's fluorescence. Okay? The light that we see is fluorescence. And then this 3rd and 4th scenarios that are shown on the diagram are those that are involved in photosynthesis. So the 3rd, that's in the red box, okay, one of them that's in the red box, is energy transfer. It's also known as excitation energy transfer. And this is where the energy absorbed by the first chromophore is passed to a second chromophore. It's just the energy that's passed. Okay? And then the 4th scenario is where we see an electron transfer happening, so that the original pigment molecule that picked up the light energy, okay, becomes oxidized, and another molecule becomes reduced. Okay, so it's these last 2 scenarios that are part of photosynthesis, or make up the light harvesting reactions of photosynthesis. So this next slide is definitely one that I used in BIOC3005 to basically illustrate that there are 2 types of pigment protein complexes in the thylakoids, and they make up what are known as photosystems. Okay, so photosystems are large systems made up of antenna complexes or light harvesting complexes, and also reaction centers. So the light harvesting complexes take in the light energy from the sun. They pass that light energy from one antenna complex to the next, and that's what's illustrated in the right-hand cartoon on the slide. So that bouncing red line illustrates the light energy coming in from the sun, hitting one antenna complex, being passed, that energy being passed to another, to another, to another. That energy transfer is exciton energy transfer. Okay? No transfer of electrons. It's exciton energy transfer. Then, when the wavelength of light is of a particular wavelength, it will hit specialized chlorophyll A molecules that absorb that wavelength of light specifically. Okay? And that happens in a reaction center. That's illustrated in the diagram as well. And it's in the reaction center that we see a separation of charge. That's where we see the transfer of an electron. Up until then, no. Exciton energy transfer only, but in the reaction center, that's where we see the electron being donated and accepted by another molecule. So what are the players in these light reactions of chloroplasts? What are the complexes that are embedded in those thylakoid membranes? The 2 photosystems are known as photosystem 2 and photosystem 1. Again, I talked about these at level 2. This is where we see the specialized chlorophyll molecules that will then donate electrons. So in photosystem 2, those are the P680 chlorophylls. In photosystem 1, that's the P700 chlorophylls. What I didn't talk about at level 2 were the major proteins in these reaction centers. and they are the D1 and D2 proteins. Okay? And then in photosystem 2 and in photosystem 1, it's PSAA and PSAB. Those are the major proteins. Okay? So these are the reaction centers, okay, that are found in the photosystems. But associated with those photosystems, remember, are also antenna complexes, light harvesting complexes, funneling the light energy into the reaction centers. And then there's also a cytochrome complex. It should sound familiar because there's a cytochrome complex in the mitochondrial electron transport chain, right? So in the chloroplast, it's the cytochrome BF complex, so different types of cytochrome compared to what's found in the mitochondria. In this complex, there's also an iron-sulfur protein known as the risky iron-sulfur protein. So a great complex as far as electron acceptors and donors, right? So we've got the heme, the iron in the heme groups. We've got the iron-sulfur protein, the risky iron-sulfur protein. And then there are mobile electron carriers, just as we saw in the mitochondrial electron transport chain, also in the chloroplast electron transport chain. Plastoquinone, so just add a plasto in front of it, designating that it's found in the chloroplast. Quinone, remember, is found in the mitochondria. It's lipid soluble, so it works within the plane of the thylakoid membrane. It carries 2 electrons. And then plastocyanin is a mobile electron carrier found on the luminal side of the thylakoid membrane. So remember, that's the space that's formed when the thylakoid membranes wrap on itself. Okay, so that space on the luminal surface, that's where plastocyanin works. Cyanin, you can tell from its name, it's a copper-containing molecule, a single electron carrier. And then ferredoxin is located on the opposite side of the thylakoid membranes. It's found on the stromal surface of the thylakoid membranes. Again, you can tell from its name, it's an iron-sulfur containing compound, carries 1 electron at a time. And then there's a second electron carrier on the stromal side of the thylakoid membranes that you would not have heard about at level 2. And this is the ferredoxin NADP oxidoreductase, or FNR, and we'll talk about it later in the lecture. So again, on the stromal surface of the thylakoid membrane, it's a flavoprotein, as you would guess, because it's got an NADP plus prosthetic group associated with it, as well as iron in the ferredoxin part of the name. All right, so putting all of those complexes together, all of those mobile electron carriers together, that's what's shown in this diagram. So electrons are transferred from the P680 specialized chlorophyll A molecules to plastoquinone, and that's illustrated in the inset diagram. So the close-up of photosystem 2, there's the P680 chlorophylls getting bombarded by light energy, okay? And that's where we see the electron transfer happening in the reaction center, and the electrons move through photosystem 2 into the plastoquinone pool. Basically, we talked about the quinone pool in mitochondria. We can talk about the plastoquinone pool in chloroplast. Now, when that electron is given up by the P680 chlorophylls, that means there's an electron hole in those chlorophylls, okay? That means that they're not photochemically competent anymore. They can't play anymore in light harvesting. So they have to be re-reduced. Where do the electrons come from? Well, the immediate electron donor is what's known as Z. Okay? Z is actually a tyrosine residue, so the amino acid tyrosine, one of the tyrosines in the D1 protein, one of the major proteins in the photosystem 2 complex. So Z donates an electron to the P680 chlorophylls. P680 chlorophylls are now photochemically competent again, but now Z is compromised. Where does Z get its electrons to be re-reduced? They come from the oxidation of water. So remember, photosystem 2 is where the water splitting complex is found, Okay? So water is split, electrons are given up, those electrons are acquired by Z. Okay? And notice that, of course, when water gets split, protons are released. So we've got protons building up in the thylakoid lumen. All right, so if we go back to the reduced side of photosystem 2, we've now got plastoquinone as plastoquinol, the reduced form, and that compound, remember, is a mobile electron carrier that works in the plane of the thylakoid membranes, and that's illustrated in the lower diagram where we see the PQ-PQH2 pool in the thylakoid membrane. Electrons will be donated from the PQ pool to cytochrome B6F. Now when that happens, 2 protons are moved across the thylakoid membrane. For each electron that is moved through photosystem 2 in the PQ pool. Okay? And from cytochrome B6F, the electrons are transferred to the second mobile electron carrier that we talked about, plastocyanin, which again works on the luminal surface of the thylakoid membrane. Okay? So for each pair of electrons moving through the photosystem 2, PQ pool, and cytochrome B6F, 4 protons are moved across the membrane, okay, for each pair. All right, so now we've got electrons in plastocyanin are oxidized. Now, how do they become oxidized? It's through, again, the absorption of light energy. So light energy comes in, hits the P700 chlorophyll molecules in photosystem 1. The electrons from these specialized chlorophyll A molecules then move through the photosystem 1 complex until they reach ferredoxin, and then they move on to reduce FNR. But now we've got the P700 chlorophyll molecules in a photochemically incompetent state. In other words, the electron hole in the P700 chlorophyll A molecules has to be filled. And that's where we see plastocyanin coming back into the picture. Plastocyanin will donate electrons to the P700 chlorophylls in photosystem 1. So from ferredoxin on the stromal surface of the thylakoid membranes, we see electrons being transferred to FNR, and from FNR we see NADPH being reduced to NADPH. So NADP plus to NADPH, and 2 single electron steps are required to fully reduce NADP plus to NADPH. So we're seeing a buildup of protons from the oxidation of water and from the activity at the cytochrome B6F part of the electron transport chain. And so this establishes a proton gradient across the thylakoid membrane, where the pH in the thylakoid lumen can be 5 typically, whereas the pH in the stroma is about 8. So a 1,000-fold difference of protons across the thylakoid membrane, a very strong gradient to drive the production of ATP through the ATP synthase. The process is known as photophosphorylation, to distinguish it from oxidative phosphorylation that happens in the mitochondria. So I'll leave a question with you. Does this process happen at night? Does photophosphorylation happen at night? The ATP synthase that's found in the chloroplast is essentially identical to that in the mitochondria, except for one component, and that is a subunit that's found in the CFO region The CFO region is the transmembrane part of the ATP synthase that's found in the chloroplast thylakoid membranes. Subunit 4 has no clear role. Subunits 1, 2, and 3 are analogous to subunits A, B, and C in the mitochondrial ATP synthase. Okay? We don't really know what subunit 4 is doing in the chloroplast ATP synthase. Okay? We just know that it's there, perhaps some function, some structural role. Okay? The CF1 region of the chloroplast ATP synthase is identical to that of the mitochondrial ATP synthase. It's the part that projects into the stroma in the case of the chloroplast, but the structure itself, the subunits are the same number of subunits, and the interface of the alpha and beta subunits is where ADP, an inorganic phosphate, is converted to ATP. So the mechanism is the same. Just a word of caution, many people think that the C means chloroplast. It doesn't. CF is coupling factor. That was the original name of the components of the ATP synthase, coupling in the chloroplast, coupling factor O and coupling factor 1. Okay. So what we've been talking about so far is known as linear electron flow. Okay. So electron flow from photosystem 2 through the cytochrome B6F into photosystem 1, and that involves those 3 mobile electron carriers. Okay. And actually 4 if we count that FNR. Okay, so plastoquinone, plastocyanin, ferredoxin, and FNR as all of the mobile electron carriers. Right? What I'm going to talk about now is a process that you probably haven't heard about before, and this is known as cyclic electron flow and photophosphorylation. In cyclic electron flow, as shown in the diagram, photosystem 2 is not involved, which means there's no water splitting happening, and that also means that protons don't build up to the same extent in this pathway as they do in linear electron flow. In this pathway, electrons going through photosystem 1, okay, get to ferredoxin, as in the linear pathway that we've already talked about, but from ferredoxin, they do not reduce FNR. Instead, they reduce what's known as ferredoxin PQ complex, okay? Or in the full name, ferredoxin PQ oxidoreductase complex. Okay? And from there, electrons are transferred into the PQ pool. And of course, from there, electrons are donated from the PQ pool into or to cytochrome B6F, then plastocyanin, back through photosystem 1 to ferredoxin, and that's the cycle. That's cyclic electron flow. ATP is still produced because remember, at cytochrome B6F, there are protons being transferred across the thylakoid membrane from the stroma to the lumen, but the gradient isn't as high because there's no water splitting. However, there's no NADPH made because ferredoxin is not reducing FNR, which then reduces NADP+. Instead, ferredoxin reduces the ferredoxin PQ oxidoreductase. So why would plants want to do this? NADPH and ATP are used in a number of biosynthetic pathways, including carbon fixation that we're going to be talking about in the next lecture. Okay? So plants have to be able to balance how much NADPH they need, how much ATP they need for all of their biosynthetic processes, because basically a lot of the ATP in plants comes from the chloroplast. Yes, it comes from the mitochondria as well, but there's lots of reactions happening in the chloroplast that are of benefit, if we can think about it that way, to the plants, like making carbohydrates for growth and development, right? So cyclic electron flow is involved in regulating the amount of ATP and NADPH that a plant makes. The rate of cyclic electron flow is much less than the linear or non-cyclic photophosphorylation that we've talked about. It's less than 5%. Okay? But it's extremely important when there's changing light conditions that a plant has to deal with, or changing metabolism that the plant has to deal with. And also, just for interest, the ferredoxin PQ oxidoreductase has not been characterized. We know it's there. We really don't know all of its structure or actually how it works at the molecular level. Okay, another aspect of plants that we need to think about, because plants cannot move when it's bright and sunny. They cannot move into the shade. They have to stay put and deal with the light energy that's hitting them. When it's cloudy, they can't try to find a sunnier space. They have to deal with the light energy that's hitting them. So how do they do with that? Yes, cyclic and non-cyclic electron flow, but there's also underlying structural changes that happen depending on the amount of light that hits a plant, that hits the leaf of a plant, that hits an individual chloroplast and an individual thylakoid membrane within the chloroplast of a plant. Okay? So we've already talked about there being two types of thylakoid membranes, the stacked and unstacked, the granule membranes or the stromal membranes. You can also call them oppressed for stacked, unoppressed for unstacked. Okay? All of those three different terms for the two different types of membranes can be used. We see a difference in the content of those thylakoid membranes depending on whether they're stacked or unstacked. In the stacked regions of the thylakoid membranes, we see photosystem II complexes and their light harvesting complexes, so LHC2s. Okay? We also see cytochrome B6F complexes. So the electron micrograph shows very nicely the stacked granule regions, very densely stained in the electron micrographs, but then the stromal thylakoids that connect adjacent granule stacks. If we look at the cartoon at the bottom of the slide, then we can see where the photosystem II complexes and the cytochrome B6F complexes are located in the stacked regions of the thylakoid membranes, and then the associated light harvesting complex IIs with photosystem II. In the unstacked region of the thylakoids, or the non-oppressed regions, that's where we see photosystem I complexes and the ATP synthase, which makes sense because the CF1 regions projecting out basically make it impossible for the ATP synthase to be in the oppressed regions. There's not room for it, for those complexes to be there. So ATP synthases, photosystem I, are in the non-oppressed regions, as are some cytochrome B6F complexes. I think this diagram is a good one because it illustrates that photosystem I and photosystem II are not present in equal amounts in the thylakoid membranes, and the amounts change depending on light quality. With the typical textbook illustrations of the linear electron flow, we see photosystem I, cytochrome B6F, and photosystem II, right? But that isn't what happens in real life, okay? There's not as many photosystem I's as there are photosystem II's under certain light conditions, and vice versa, right? It's not a one-to-one correspondence. And it's the same with cytochrome B6F. Cytochrome B6F is associated with both of the photosystems, so it's not necessarily one-to-one-to-one ratio. Okay? So, how does this fit together with function of light harvesting? For light harvesting to work efficiently, energy has to be distributed in a balanced way between photosystem I and photosystem II. Not equally distributed, but balanced distribution. Okay? So, when we think about light energy coming in and hitting photosystem I and photosystem II, some light energy is going to be more optimal for photosystem II to absorb than photosystem I. And when that happens, the PQ pool becomes highly reduced. because photosystem 2 is being overexcited. Okay? It's collecting more of the light energy that's coming into the chloroplast because more of that light energy is favorable for the P680 chlorophylls in photosystem 2. Now, when that happens, and if we go to the top part of the diagram on the right-hand side of the slide, when the PQ pool becomes over-reduced or becomes highly reduced, that activates an LHC2 kinase. So that's illustrated in the first step of the diagram on the right-hand side of the slide. That kinase uses ATP to phosphorylate the LHC2s in the granule membranes that are associated with photosystem 2. Okay, so the LHC2s are the red structures, photosystem 2 the green structures in the cartoon. So the LHC2s get phosphorylated, and that addition of the phosphate group allows for electrostatic repulsion of the thylakoid membranes. Very, very subtle changes, okay? Very, very subtle unstacking of the thylakoid membranes. And that allows those LHC2s to move out of the stacked regions of the thylakoid membranes towards the stromal thylakoids, toward the unstacked regions. Now remember, I said in the first part of the lecture that the thylakoid membranes have a lot of unsaturated fatty acids, allowing them to be fluid, allowing these complexes to move laterally within the plane of the membrane. Clearly important here because of the light energy that comes into the chloroplast and hits the thylakoid membranes. So in this scenario now, the LHC2s are directing light energy into photosystem 1 and less goes into photosystem 2, balancing that energy distribution between the two photosystems. Now, of course, there are times when the PQ pool becomes highly oxidized because photosystem 1 is becoming overexcited. So what happens then? Well, then the LHC2 kinase loses activity and the LHC2 become dephosphorylated by a specific phosphatase. So that's shown at the lower part of the diagram. And that allows the LHC2s to migrate back to the granule stacks, to the oppressed thylakoid membranes, directing more light energy to photosystem 2, less to photosystem 1, and we see a rebalancing of the energy distribution. So this beautiful process, okay, allows light energy to be balanced between the two photosystems, and it also allows very efficient light energy harvesting. And speaking about efficiency and light harvesting, we can actually measure it, and it's known as quantum yield. So quantum yield, and that's designated by the Greek symbol phi, is a measure of photochemical efficiency, where phi is the number of products that are formed through the photochemical process per number of quanta, so light energy packets, absorbed. And where phi is 1, that means for every photon absorbed, there's a chemical product produced. Okay? When values are less than 1, then that means decay processes are happening. For example, fluorescence is involved, and the light energy is not used in photosynthesis, is not used in photochemistry. What we see with plants is that when they're working under optimal conditions, photosystems have a phi of 1. They're very, very efficient at harvesting light energy and converting it to chemical energy. And then just as a wrap up for the lecture, at the bottom of the slide is the stoichiometry of linear photophosphorylation, where we see 8 photons being involved, 4 hitting photosystem 2, 4 hitting photosystem 1, along with water being split, ATP being generated from ADP and inorganic phosphate, and NADPH, reducing power, being formed from the reduction of NADP+. And I'll leave it there. Thank you.